Chapter 21, it's about the lymphatic system. So the lymphatic system is also known as the immune system because both of them, they have essentially similar functions. So the immune system, it's part of our system of defense mechanisms that has a large population of cells, many tissues and organs, that all of them help to recognize foreign substances that we know as antigens. And they act to neutralize and destroy these foreign substances. Now, one difference between the lymphatic organs and the lymphatic tissues is that the lymphatic organs are surrounded and protected by a fibrous capsule, so they have a defined structure, and then the lymphatic tissues do not have such a capsule. And because of this, the lymphatic tissues do not have a defined structure. So this is one of the key points then that you need to understand, that lymphatic organs do have a fibrous capsule, while lymphatic tissues do not have a capsule. Now, the functions of the immune or the lymphatic system are various. So one of these will be to recover fluid. Remember that when the capillaries bring fluid into the extracellular space so that the cells in our body can grab oxygen, nutrients, hormones, etc., these capillaries leave a certain amount, and then they reabsorb 85% of the fluid that they deliver. But the other 15%, it will be recovered by the lymphatic system. And we have between 2 to 4 liters that are then delivered into the lymphatic system so that the lymphatic system can return this fluid into the circulatory system. And the composition of this fluid, lymphatic fluid, is similar to the one of the plasma. Okay, and the other function of the immune system, of course, will be defense or immunity. And when these cells of the immune system find anything that it doesn't look right, like presence of cancer cells or an antigen or a pathogen that they don't consider their own, then these cells of the immune system will attack these foreign substances or these damaged cells or the cancer cells, and they help to prevent damage to our body. The other function of the lymphatic system is to pick up lipids. So within our intestines, whenever we ingest lipids, they are broken down into small molecules by the enzymes that are released by the pancreas, and then the intestinal cells in the jejunum and in the ileum start absorbing these lipids. And these lipids are large, so they cannot directly be absorbed by the intestinal cells. So they are broken down first, then they are absorbed, and then the lymphatic vessels that we have within the enterocytes or the cells of the small intestine pick up these lipids, and then they bring it into the circulatory system. Now, these specific lymphatic vessels that we have within the small intestines are called lacteals. So, the other parts of the lymphatic system will be the lymph, which is the recovered fluid, the 15% of the fluid that it is not picked up by the capillaries. And then we have these lymphatic vessels that are basically similar to blood vessels, but they are called lymphatic vessels, and they will pick up this lymph and then they will transport the lymph. So the other components, the lymphatic tissues. So the lymphatic tissues, as we said before, they don't have a capsule, and there is this scattered tissue of the lymphatic system that has nodules or aggregates of lymphocytes and macrophages that helps to protect many organs in the body. For instance, we have lymphatic tissues surrounding the stomach and also surrounding the intestinal cells. Now, the lymphatic organs, as we said before, they have a fibrous connective tissue or a capsule that provides a specific structure. And within these lymphatic organs, we also have aggregates of lymphocytes that are called the nodules. And within these nodules, if one of the antigens that is circulating through the lymph or one of these abnormal cells that is circulating through the lymph find a nodule, these nodules with its cells, with the lymphocytes, it will detect this antigen and then they will start doing clonal expansion through mitosis, so you can have more prepared lymphocytes to attack these foreign substances or cells. Characteristics of the lymph. The lymph is a fluid, it's colorless, it's clear, and it's similar to plasma, but it has less protein. And the origin of the lymph will be the extracellular fluid that wasn't being picked up by the capillaries. And depending on which area of the body is where you have this lymph is going to be the chemical composition. So in the intestines, the lymph fluid is going to be rich with lipids because you are absorbing lipids. Now, the lymphatic fluid before the lymph nodes, which are basically collections or little organs of the lymphatic system, which are collections of lymphocytes, before the lymph reaches these lymph nodes, if someone has an infection, you're supposed to have bacteria, for instance, or viruses within this pre-filtered lymph. And then after the lymph leaves these lymph nodes, it's supposed to be filtered or clear up of bacteria. So then the chemical composition will vary. Another name for the lymphatic vessels is also known as lymphatic capillaries or terminal lymphatics. They are found everywhere in our body, just similar as for the capillaries of the circulatory system, and as well as these capillaries of the circulatory system, these lymphatic capillaries are not found in cartilage, cornea, bone, and bone marrow. The capillary of the lymphatic vessels, the lymphatic vessels, they have only one layer of cells, which is the endothelial cells, and then they have an extension, these endothelial cells, that form like little flaps or valves that overlap to each other, looking like roof shingles. Lymphatic vessels are not continuous. They have a closed end, and they're usually, like I said before, found in the capillary beds because this is where the fluid of the capillaries is drained into, and then the lymphatics will pick it up. Now the fluid, the lymph will enter through a gap in between these endothelial cells, and they have these large gaps that even bacteria can enter into these lymphatics. So this diagram or this cartoon shows you the capillaries and the capillary beds, which are these in red and blue, and also it shows you these lymphatic vessels or lymphatic capillaries, which are these ones in green. As you can see here, they have a closed end, and then they have large gaps that allows the entry of this lymph, and also bacteria or foreign cells can enter through the lymphatics. If you make a magnification of one of these lymphatics, you can see how they have within their walls only one cell, a squamous cell, which is the endothelium of the capillaries, and then they have these extensions that form like little valves or flaps. Now, as the lymphatics move away from the capillary beds, they form larger lymphatic vessels. And some of these large lymphatic vessels can have the three layers of the large blood vessels. They can have a tunica interna with the endothelium and valves. They will have a tunica media that is made of elastic fibers and smooth muscle cells, and a tunica externa that it is made of connective tissue. And as they form larger vessels, they will start draining into lymph nodes so that the lymph nodes can clear up the lymph. This is the histology, the microscopic view of the valves within the lymphatic vessels. So they are regularly closed, and once the lymph enters from distal regions into proximal regions, this lymph will flow from proximal to distal. And then as the fluid expands these walls, they will close these valves, preventing backflow of the lymph. So when large lymphatic capillaries merge, they will form trunks. And when the trunks will merge, will form ducts. And we have 6 major lymphatic trunks that drains major portions of the body. We have jugular lymphatic trunk, we have subclavian, we have bronchomediastinal, we have intercostal, intestinal, and lumbar trunks. Jugular, subclavian, and bronchomediastinal are pretty much close to each other. Jugular is found in the neck, subclavian is just in between the neck and the shoulder, bronchomediastinal is found in between the lungs and the heart, intercostals are found in between the ribs, intestinals will be within the abdominal cavity together with the lumbar trunks, and the intestinal, they will drain the lymph from the intestines, lumbar trunks from the pelvic region, and also they will drain the lymph from the legs. Jugular, they will drain the lymph from the face and the scalp. The subclavian, they will drain the lymph from the arms, and bronchomediastinal will drain the lymph from the lung region and the heart region. Now, some of these trunks will merge, and then they will form these major ducts. And we have 2 major collecting ducts. We have right lymphatic duct that will drain the area that is above from the diaphragm on the right side, including the right half of the head. And this right lymphatic duct will drain into the right subclavian vein. So that is the connection then between the lymphatic system and the circulatory system. So the lymph has to return into the circulatory system. So the lymph will be drained into veins, and the right lymphatic duct will drain into the right subclavian vein, while the thoracic duct, the largest duct that we have, because it drains basically everything below the diaphragm and everything on the left side of the body above the diaphragm, including of course the arm, including the head, and the left side of the head and the neck. So that's why this is the largest, the thoracic duct. So the thoracic duct will drain into the left subclavian vein, and then the subclavian veins will drain into the brachiocephalic vein, and then they will drain into the right atrium. So again, this is just to emphasize how the lymphatic system, it is connected to the cardiovascular system, so as you can see here within this figure. So anything that the that the capillaries of the circulatory system, the capillary beds, they don't pick up the excess of fluid, the 15%, it will be drained into these lymphatic vessels that they will form trunks, and then the trunks will drain into the ducts, and the ducts, they will drain into the veins of the circulatory system. The thoracic duct, which is here found in the left side of the body, will drain into the left subclavian vein, while the right lymphatic duct, which is found here on the right side of the body, will drain into the right subclavian vein. And then, as you can see here, we have within the body a lot of lymph nodes. These lymph nodes are little organs of the lymphatic system that helps to drain the lymph, so that if anything goes wrong, like let's say that this person has an infection here in the foot, these popliteal lymph nodes will help to prevent these bacteria that are infecting the foot from going into proximal regions, so from spreading above the knee. So these lymph nodes will become enlarged when someone has an infection in the foot, and so on and so forth. So in this diagram, it shows you the lymphatics of the thoracic region. So we have lymphatics within the breast, and these lymphatics of the breast, they will drain into these axillary lymph nodes. And then these axillary lymph nodes will drain, because this is the right side of the body, will drain this lymph into these lymphatic vessels that will drain into the right lymphatic duct. So this is important because in the case of cancer cells, for instance, if a female has breast cancer on the right breast, these cancer cells can move from this region, from the breast, they can move into other parts of the body using these lymph nodes. So they will migrate or metastasize through these lymphatic vessels. They will reach these axillary lymph nodes. These axillary lymph nodes will swell to try to kind of stop the spreading of the cancer cells, and then they can go into the rest of the body. And this is important at some point, because let's say that if this, in this case, this female will have cancer on the left side, on the left breast. These lymph nodes on the right side will not swollen, only the ones on the left. So unilateral inflammation or swelling of swollen lymph nodes pinpoint only to a unilateral problem. Okay, so here we have the drainage of the lymph within the thorax. So here we have these lymphatics, intercostal lymphatics, that they will drain, depending on the site, here into this thoracic duct, which is this large thoracic duct, or this duct, or they can drain into the right lymphatic duct. So it is not very clear in here, but of this lymphatic duct, the right lymphatic duct, but at some point this is the largest, the thoracic duct. Why? Because it drains anything below the diaphragm, like the intestines or legs, etc. And also this thoracic duct will drain the lymph from left arm, left side of the neck, left side of the head. And then it will drain here the left thoracic duct into the left subclavian, and the right lymphatic duct into the right subclavian. And you can see also here, now that we have this detail, you can see these lumbar ducts. We have the right and the left, and the intestinal ducts that drains into this expansion. This expansion in the thoracic cavity is called the cisterna chyli, or chili. And then from there, the lymph will continue up, and then it goes into the thorax. Okay, so we talked then about the vessels of the lymphatic system. Now let's talk about the lymphatic system cells. So we have several different cells that helps in immunity or defense. So we have the NK cells, which are basically the immunological surveillance. They are like patrolling the body. They're constantly circulating throughout the blood vessels to find if anything goes wrong. And these are lymphocytes, but they are called natural killer cells, or NK cells, because once they find something abnormal, like a bacteria, transplanted tissue, host cells infected with viruses, or cancer cells, these natural killer cells are like snipers. It will kill without investigating anything. It will kill directly the cells. Okay, that's why they're called the NK. Now, the other type of lymphatic cells will be the T lymphocytes, or T cells, and they are in charge of producing cellular immunity. So if someone has an infection within the cells, these T lymphocytes will destroy these cells. And these T lymphocytes, they are produced in the bone marrow, but they mature in the thymus. And we have 4 subclasses of T lymphocytes. We have the CD4, or helper cells. We have the CD8, or cytotoxic. We have suppressors, or regulatory, and we have memory. And the functions of them we will talk about in another segment of this presentation. Now, the B lymphocytes, or B cells, they produce humoral immunity. So if someone has an infection within body fluids like the CSF, cerebrospinal fluid, within the blood, within the synovial fluid, within the urine, etc., these B lymphocytes will be in charge of protecting us. B lymphocytes are produced in the bone marrow, and they mature in the bone marrow. And we have 3 types. We have naive, the ones that still haven't been exposed to antigens. And then we have plasma cells, or activated B lymphocytes, that makes the antibodies. And then we have memory B cells. Macrophages are also part of the lymphatic cells, and these are large cells that derive from monocytes. And they get activated when they are exposed to antigens, and then they become enlarged, and then they will start phagocytosing, or eating up tissue debris, dead neutrophils, bacteria, and foreign matter. So they are kind of the janitor cells. And one way they help in producing defense is once they find an antigen, they engulf this antigen, they make pieces out of them, and then they present them into the surface using some molecules that are called MHC, or major histocompatibility complex. And then the T lymphocytes will be attracted by these fragments of antigens, and they will become activated. So the macrophages, together with other cells, are known as antigen presenting cells, or APCs. And this is a macrophage that it is attacking bacteria. So in order to engulf these bacteria, these macrophages extends the cytoplasm and the plasma membrane, forming what we call false feet, or pseudopods. Pseudo means false, pods means feet. So all of these in green are bacteria, and this one in purple, this large cell, is the macrophage. Another antigen presenting cells are the dendritic cells. These dendritic cells are found in many parts of the body, like the epidermis, mucous membranes, and lymphatic organs. And some of them, they have different names depending on where they are located. But in general, what they do is similar to the macrophages. They just find antigens in these places, and then they put it into their plasma membrane, and then they present it into the T lymphocytes so that they can become activated. So the function is basically to alert the immune system that the pathogens have breached the body surface. So this will be one of the first cells that the bacteria or viruses or other pathogens can encounter. So if you have a cut, and this cut in the mucous membrane or in the skin was infected with bacteria, the dendritic cells become activated. Reticular cells are similar to dendritic cells in function, but these are stationary dendritic cells that are located within lymphatic organs, especially within the lymph nodes. Okay, so we talk about the cells. Now let's talk about the lymphatic tissues. So lymphatic tissues are known also as lymphoid tissues, and they are collections of lymphocytes or nodules of lymphocytes located within the connective tissues of mucous membranes and various organs. Now, when this lymphatic tissue is located within the mucosas, is also known as MALT, M-A-L-T, as you can see on the bottom of the slide, mucosa-associated lymphatic tissue. And diffuse lymphatic tissue is the simplest form of lymphatic tissue. The lymphocytes are scattered, they are not clustered, and they are found in areas of the body that are in close contact with the environment. Examples of areas where we have this lymphatic tissue, diffuse lymphatic tissue, will be within the respiratory mucosa, digestive system mucosa, urinary system, and reproductive tract. Another type of lymphatic tissue will be the lymphatic nodules or follicles. And in here we have a denser population of lymphocytes in a specific area. And we also have macrophages. And we have them within the lymph nodes, the tonsils, and the appendix. And the aggregated lymph nodules, they can take a specific name in different areas. So for instance, in the small intestine, in the ileum, they will be called the oh my gosh, let me remember, guys, sorry. Peyer's Patches, P-E-Y-E-R-S, Peyer's Patches. So this is an example of lymphatic nodule. All of these little dots that you see here in purple will be the lymphocytes that were stained with the stains that are used in histology. And this part that you can see here are the intestinal bullous. So this is part of the intestine. So here it's another picture of the small intestine, specifically will be within the ileum, and these will be the Peyer's patches that you can see here, these ones. This in your book, they just or in the PowerPoint, they just put it in blue so you can see it, but actually this will be kind of a purple color. Okay, they just changed the color for you to know where exactly it will be located. Okay, and here's the epithelium of the intestines. Okay, so now for the lymphatic organs, so they are anatomically defined because they have a capsule, fibrous capsule, and because of this, these organs are definitely separated from the neighboring tissues. So we have a subdivision of the lymphatic organs, primary lymphatic organs and secondary lymphatic organs. Primary lymphatic organs will be the place where the cells of the lymphatic system are produced, like or trained, like the red bone marrow and the thymus. And the secondary lymphatic organs will be the area or the organs where these lymphatic cells, the lymphocytes, can live on, or the antigen presenting cells. So which ones are the primary lymphatic organs? Will be the bone marrow, the thymus. And which ones will be the secondary lymphatic organs? The lymph nodes, tonsils, spleen. And within the bone marrow, we will produce both B and T lymphocytes. And specifically, these cells will be derived from either the monocyte colony forming unit for the macrophages or the lymphocyte colony forming unit. So we have a lymphoid lineage for the lymphocytes within the stem cells that are found within the bone marrow. And once the bone marrow produces these lymphocytes or these macrophages, they will exit through these sinusoids in the bone marrow, and then they will enter the circulation. So this is to show you then how these cells are produced, the T and the B cells, both of them in the bone marrow. But the B cells will mature in here, while the T stem cells will come here into this thymus, and then they will be trained, and then from there they will go into the circulatory system. Now at some point, the thymus will train our T cells for as long as we have a thymus. So our thymus, it is present when we're born. If we don't have a genetic disease, it will be present, and it will disappear like around the puberty years, 14, 15, 16 years of age. So it will involute, it will regress, because by that time we have been exposed to so many antigens in our environment that we don't need a training school for the T lymphocytes. We're supposed to be fully immunocompetent at some point, and then the thymus will disappear or regress. And then the B cells and the T cells, once they are mature, they can go and populate the lymph nodes, the spleen, the tonsils, or other organs. And this is just to show you how here within the bone marrow, we have these immunocompetent cells. And then through these openings in the sinusoid, these fenestrations, we will have the entry of these cells that we have produced within the bone marrow. Now let's talk about specific characteristics of the secondary organs of the lymphatic system. So the thymus, it is member not only from the lymphatic system, but also from the endocrine system. This will receive the developing lymphocytes, the T lymphocytes, and then upon the action of hormones like thymosin, we will have this maturation of these T lymphocytes. Thymus has this specific location within the superior and anterior mediastinum. So it is located above the heart and between the sternum and the aortic arch. And it has kind of a bilobe shape. I cannot tell you specifically what is the shape, but it has two lobes. Okay, so it has the right and the left, and it's supposed to degenerate or involute with age. If after puberty this organ does not involute or regresses in size, they might be a cancer, lymphoma. Okay, so this thymus is surrounded, because it's an organ, by a fibrous capsule, and this fibrous capsule will become internal, so it will go inside the thymus, and it will separate the gland into several lobes. And this fibrous capsule, as it's going inside, it will be forming septa or trabeculae. And Within the lobes, we will have an area that it is superficial, that is closer to the capsule, that is called the cortex, and the most internal region will be called the medulla. Within the medulla is where we have the T lymphocytes. And we have epithelial cells sealing off the cortex from the medulla, and they will form the blood-thymus barrier. This is important because it will help to maintain signaling molecules like the hormones thymosin, thymopoietin, thymulin, interleukins, and interferon from just diffusing at a gradual rate, so that they can help to mature these T lymphocytes gradually, not so fast. So at some point, the thymus, if you see it, it might have like kind of a triangular shape or conical shape. And you see here the left lobe and the right lobe. And all these little groups that you see here will be the trabecula. So if you make a section of one of these lobes and you magnify it, you will be able to see this capsule and then this epithelium. And then in here we have the cortical epithelial cells, which are forming part of the cortex. And then here we have the blood-thymus barrier. So this is where we have the thymosins and all these hormones activating these T lymphocytes, or training these T lymphocytes, not activating them, training them. And here we have dendritic cells that supports all of this tissue. And we have here this tissue that it is specific for the thymus, that is called the thymic corpuscle. This is the actual micrograph of the thymus. So you can see here the cortex, the medulla, and the lobules. This will be the trabeculae that separates the thymus into little segments or lobes. And if you see a greater magnification of the thymus, you can see these Hassell, Hassels or thymic corpuscles. And all of these purple cells that you see here will be the T lymphocytes. Okay, another organ will be the lymph nodes. The lymph nodes will be the most numerous organs within the lymphatic system. We have like around 450 and in the adults, and then they have the purpose of filtering or cleansing the lymph, and will activate the T and the B cells. So they have a shape like a bean shape, and they have an indentation like pinto beans that it is called the hilum. And within this fibrous capsule, we also have the extension internally that is also known as the septum or trabecula, that will divide the interior of the lymph nodes into compartments. And within these compartments, we will have reticular fibers and reticular cells. So this is the lymph node. So this will be the convexity of the lymph node. This will be the concavity. So this is the hilum, and this will be the cortex that you can see here. And it has a middle region that is known as the medulla. And within the cortex, we have these germinal centers. If you magnify one of these regions within the lymph nodes, you will be able to see the capsule, which is this. And then you will be able to see these germinal centers or lymph nodes, lymphatic nodules. So in here, within these germinal centers, we will have these lymphocytes that, as the lymph is passing through, they will help to detect anything wrong within the lymph. If they find viruses, bacteria, antigens, cancer cells, these germinal centers will grow, and that's how we will have swollen lymph nodes, and then hopefully they will be able to clean the lymph that is passing through here. So the parenchyma within the lymph nodes is divided into the cortex and the medulla, or the tissue. Another name for tissue will be parenchyma. And the germinal centers will be where the B cells will multiply and differentiate into plasma cells. So if your B cells find an antigen that it is not part of our body, so if it's a foreign antigen, the B cells will detect it, and then they will start become activated. So the naive B cells will become activated, will become plasma cells, and they will secrete antibodies that will bind to these antigens, neutralize these antigens. And it will work together with other cells to set an immune response. Now, within the cortex, we have the arrival of several lymphatic vessels, afferent lymphatic vessels, arriving. Think about arriving as afferent, and they will arrive into the convex surface of the lymph nodes. And only in the hilum we will have 1 to 3 efferent lymphatic vessels. So there is more lymph that arrives to the lymphatic nodes than the efferent or exiting lymph. So if you think about it, like you can see it in here. In this case, we have 1, 2, 3, 4, 5 afferent vessels that brings the lymph, as you can see here, brings the lymph into the lymph nodule, and only we have 1 efferent here in the hilum. So if you think about this, you will create a bottleneck. Is this important? Yes. Why? Because as you are receiving a lot of lymph within this lymph node, and not as many can exit through these efferent lymphatic vessels, you give time for this lymph to be exposed to these lymphocytes that are found here within the germinal centers to see or to find if anything it is wrong. Okay, so in this case, if you stop the flow a little bit of this lymph, you can find cancer cells much easier. You can find bacteria, viruses, any pathogen, or any antigens that are important. So if you magnify this area, you can see these trabecula and the lymphocytes and the reticular fibers, which are these ones that you can see, and the lymphocytes trapped within these reticular fibers. So the lymphocytes will be more or less a little bit static in there, waiting for the lymph to flow from these lymphatic vessels or this area into here. And sorry, this is the trabecula. Okay, so let's talk about the location of the lymph nodes. So we can have it in the neck, and they will be called cervical lymph nodes. And we have a deep and a superficial group in the neck. The deep group runs deeper to the sternocleidomastoid muscle, kind of close to the trachea, and the superficial is found superficial to the sternocleidomastoid. And they will monitor the lymph coming from the head and the neck. Now, we also have these lymph nodes within the face, and also within the area just below the mylohyoid muscle, in between your body of your mandible. They will help to detect also these lymph nodes that will drain actually into the deep or the superficial cervical lymph nodes. They will detect any infections within the oral cavity or within our teeth. Now, the axillary lymph nodes are located within the armpit. They will receive the lymph from the upper limb and in females from their breast, and they will help as well to detect any problems within these areas. And if anything is wrong, you expect to have inflammation of these lymph nodes. Inflammation of lymph nodes is called lymphadenitis, and at some point, swollen lymph nodes, guys, can be tender or non-tender, so they can be painful or not painful. Usually, if they're painful and they're swollen, it pinpoints to an infection. If they are swollen but are not painful, it pinpoints to cancer. But you cannot tell that someone has cancer unless you have a biopsy or you have studies to tell that, cytological studies to tell that someone has cancer. Now, thoracic lymph nodes are internal lymph nodes. In this case, they are located within the thoracic cavity. They are located more likely in between the lungs in the mediastinum, and they receive the lymph node from the mediastinum, the thoracic wall, the lungs, and the airway. So if someone has cancer in the lungs, let's say, the first lymph nodes that will become swollen will be the thoracic lymph nodes. If there is a way to tell when these lymph nodes, directly with physical inspection, that these lymph nodes are swollen, no, there is not, because they are located in the area between the lungs. So this is why it's hard to detect at some point cancer within the lungs. But if someone has cancer within the breast, although you might find it late at some point, but if you have a swollen lymph node within one of the axillary areas, well, it's much easier to detect than a swollen lymph node within the thoracic cavity. Similar for the abdominal lymph nodes, the intestinal lymph nodes, okay? Now, the abdominal lymph nodes, they are located within the posterior abdominal pelvic wall, and then they drain or cleanse the lymph coming from the urinary reproductive systems. When these lymph nodes of the reproductive system will be detecting problems of the internal organs of the reproductive system. The intestinal and mesenteric lymph nodes are located within a membrane that is called the mesentery that holds the intestine to the back wall of the thoracic cavity, abdominal cavity, and they are adjacent also next to the appendix, which is one of the first regions of the large intestines, and are also located within regions next to the intestines. They help to cleanse the lymph from the digestive tract. Inguinal lymph nodes. They are located in the groin, and they receive lymph from the entire lower limb and also the external part of the pelvic cavity, and at some point also from external genitalia. That's why I told you, like, one difference between the abdominal lymph nodes and the inguinal lymph nodes when it comes to monitoring the lymph from the reproductive system. If someone has an infection in the reproductive system, the external genitalia in males, the penis in females, the vagina, these inguinal lymph nodes will get swollen, but not the abdominal lymph nodes, if that makes sense. Okay, now popliteal lymph nodes, they are located on the back of the knee in the popliteal area, of course. They will receive the lymph from the knee below. And this is just to show you some of the location of the lymph nodes within the area next to the large intestine. So we have here the appendicular lymph nodes, ileocolic lymph nodes in here, superior mesenteric lymph nodes, and then inferior mesenteric lymph nodes. And then this will be the transverse mesocolic lymph nodes. Are these important? Yes, of course, because as you will see in the digestive system chapter, these large intestines, they are filled inside with the feces, which are filled with bacteria, and actually we have part of the normal flora as well of bacteria in here. But if anything goes wrong in here, well, hopefully these lymph nodes can pick that up. And also one of the areas where people can develop cancer will be the colon. So that's why these lymph nodes are important. So two things can happen then when a lymph node is under the challenge by an antigen. And it can be an antigen from a bacteria, a virus, a fungi, any microorganism, or it can be an antigen found in cancer cells. So lymphadenitis will be swollen, painful lymph nodes responding to a foreign antigen. And lymphadenopathy will be a collective term for all node diseases. They can be cancer, guys, of the lymph nodes as well. Lymphadenomas, for instance. And at some point, as I told you before, usually when someone has a swollen lymph node that is non-painful, it's still lymphadenitis, but in this case, it might be that it can be caused by cancer cells. Now, why the immune system? Why, if we have these lymph nodes, why still cancer cells can go from one place to another? Why they metastasize? That's the name for migration of cancer cells. Well, because not all cancer cells display the antigens, so they cannot be recognized. They hide their identity. Okay, so tonsils are large lymphatic nodules that are located in the walls of the pharynx. This is another key point for you. Okay, so tonsils, large lymphatic nodules located in the walls of the pharynx. So the purpose of having tonsils is because they will protect us from anything, any pathogens that we inhale or ingest. Now, is this as well a perfect defense system? Well, not at all, because at some point it depends on the number of pathogens that you're inhaling and how virulent or how aggressive are these pathogens. So you can saturate these receptors that we have in the tonsils for finding antigens, and in that case, many pathogens, if you inhale them in huge amounts, they can enter into your body. And as well, I mean, it might be very little pathogens that you might inhale or ingest, but if these bacteria or these viruses are very aggressive, then you will have an infection. And then the tonsils, in some areas, they will have a deep pit that is known as tonsillar crypts that are lined with these lymphatic nodules. Inflammation of the lymph nodes, of the tonsils, is called tonsillitis, and tonsillectomy, it is basically taking them away. Tonsillectomy, it's elective surgery for some people, but definitely if the tonsils, pharyngeal tonsils, get very close to each other, that they close the airway, that's an indication for surgery. So we have names for, and I was referring to the palatine tonsils before, okay? So we have three main sets of tonsils. We have the palatine tonsils that are located on the sides of the oral posterior lateral region of the oral cavity. This is the most commonly infected, especially by Streptococcus pyogenes, the strep throat inflammation or infection that we know about. And then lingual tonsils that are located at the base of the tongue, and then the pharyngeal tonsils that are located just back on the well, a little bit inferior and back, inferior and posterior to the roof of the nose, and they are called adenoids too. So it's a single tonsil on the wall of the nasopharynx, while the lingual tonsils are paired together with the palatine tonsils. So here's the location of each of them. So what can help us to prevent infections from inhaled microorganisms will be these ones, pharyngeal tonsils. So this is the nasopharynx. What can help us to prevent infections coming from the oral cavity will be the lingual tonsil here at the base of the tongue, and also these palatine tonsils. If you make a longitudinal section of these tonsils, you will be able to see that they are embedded within this tissue of the pharyngeal walls, which is made out of muscle cells more than likely, and connective tissue as well. And then you can see these crypts. This is the opening for the tonsillar crypts. So, and then in here we have the lymphatic nodules. Just because we have this opening and these crypts, many of the food guys also get stuck in here. And then of course, stock food in here will create an infection at some point because the food gets spoiled. And you can have halitosis or bad breath because of that. Okay, the next organ will be the spleen. The spleen is found in the left upper quadrant in the abdominal cavity. It's kind of close by the stomach, and is the largest lymphatic organ. It's a single organ. The spleen has two types of tissue in its cells, in its parenchyma, in its functional cells. So it has the red pulp and the white pulp. The red pulp is called the red pulps because these sinuses, these capillaries, are filled with erythrocytes and it looks reddish. The white pulp is the area where you have concentration of lymphocytes and macrophages that surrounds the branches of the splenic artery. And as the blood is circulating through this splenic artery, the lymphocytes that are around these arteries can pick up bacterial infections or viral infections or any kind of infections in the blood. So this helps to monitor also infections in the blood, this spleen, through the white pulp. Now, the spleen, it's also the recycling center for the red blood cells. So the red blood cells, when they still are young and they are healthy, they have this biconcave shape and they have this circular shape in their disc. They're flexible, and then they can go through the reticular tissue of the spleen, which is like a net of fibers that can trap all erythrocytes. So the healthy young erythrocytes can come and go, while the old erythrocytes close by 120 days, or the ones that they have kind of odd shape because there is this genetic defect within the erythrocytes. They get trapped within the reticular tissue, and then they get destroyed and recycled. So blood production happens as well in the spleen, but in the fetus, and the white pulp will monitor any infection within the blood, and it will stabilize the blood volume through plasma transfer to the lymphatic system. So since the spleen has a lot of blood supply, so it's highly vascular, it can be vulnerable to trauma and infection. So if someone receives a hard hit on the left side of the body, like in a crash accident, or like boxers, if they receive these hits on this area, the spleen can rupture, and then the person will bleed very easily, and the person need to have, of course, this emergency surgery in which they remove the spleen, and the person will be susceptible to infections, especially infections by bacteria that is encapsulated. This bacteria that is very aggressive, and premature death. No one knows what is the cause of premature death, but this is one of the organs in the body, guys, that we can live without. The spleen is one of the single organs that we can live without. We cannot live without brain, without heart, but with without the spleen we can live. So this is kind of the shape of the spleen as well, triangular shape. This is the internal view. So you can see here the hilum. In the hilum, which is the indentation of the organs, you can see how the blood vessels come in and out and nerves come in and out. Actually, in major organs, blood vessels come in and out, and nerves or lymphatics come in and out. Now, if you make a magnification of the spleen and you look at it under the microscope, you will be able to see this large region, which is the uh, white pulp, and here you have the splenic branches, or the branches of the splenic artery. And then in here, this reddish area that you can see here is the red pulp. So in here you have lymphocytes, and then in here you have erythrocytes. And this is the magnification to show you the different areas. So this is the red pulp here, the reddish areas, and here's the white pulp. I mean, it doesn't look that white, but here you have the lymphocytes. As you can see, when you stain the lymphocytes, they look like purple. Okay, so we are done with talking about the organs of the lymphatic system. Now let's talk about the different types of immunity that these organs and cells of the lymphatic system provide us. So first, let's talk about the pathogens. So pathogens are agents that are capable of producing disease. They will be microorganisms, including viruses, bacteria, fungi, protozoans. Okay, and we have a type of immunity that is called innate. Innate because we all born with. Innate means that we born with certain things. So we all born with all this type of immunity. So we have three types of innate immunity systems that we born with that protect us against pathogens. So we all born with the skin and mucous membranes. So the skin and the mucous membranes will form the first line of defense because any pathogen will encounter first, in order to enter into our body, it will encounter with the skin and the mucous membranes. And as long as your skin and your mucous membranes don't have an injury, don't have an opening, like if you don't have a cut in your skin, or if your mucous membrane, whether it's the oral cavity, the vagina, reproductive system, or the mucous membranes in the urinary tract and or in the digestive system, they are not broken by anything, then you have a good system, the first line of defense. Now, the second line of defense that we all born with is the cells of the blood, like the leukocytes, macrophages. We're all born with antimicrobial proteins, NK cells, and with the process of inflammation, we can set this process of inflammation and fever. So all of these form the second line of defense. If the bacteria or the pathogen penetrate the skin and the mucous membrane, then they will encounter the second line of defense. And then the third line of defense, it is the adaptive immunity. Once we're born, we will develop part of this second line of defense to attack these pathogens. And this adaptive immunity at some point leaves the body with a memory. So whenever you encounter the same pathogen on a second time, you will be able to recognize it, so you will have memory, so you will be prepared to defeat this microorganism faster. One thing about the innate immunity is that it will provide a broad range of protection against a broad range of pathogens, but it will set the same response over and over again. So because of this, it will be called inspecific. And They lack the capacity to remember the pathogens. Now, we have within these innate defenses, we have protective proteins, protective cells, and protective processes. So the other name for innate defense will be non-specific, local, and it doesn't have memory. Adaptive immunity, our body has to develop a separate immunity to each pathogen. So at some point, it is important, of course, to have innate defense because that's the first one that will respond to a pathogen. But it is also important to recognize how these pathogens can damage our body, and we can set a specific defense, whether it will be less stronger than others, just to protect us. Now let's talk about the cells of the innate system. So leukocytes and macrophages are phagocytes. Why? Because a phagocyte, phago means eat, cyte means cell, will be a cell that eats or engulf foreign matter. So macrophages, which are derived from monocytes, and the leukocytes, or of its different kinds, the leukocytes, are the phagocytes. Now, another name for these leukocytes that are not macrophages, because they phagocytose foreign matter, these are called microphages, the leukocytes that are not the macrophages. So we have the neutrophils, eosinophils, basophils that are part of these macrophages, or sorry, microphages. Monocytes, they will become macrophages, and lymphocytes are not macrophages, okay, or not microphages. Lymphocytes, they don't eat, but the neutrophils, eosinophils, and basophils, they do. Details about the neutrophils. Well, the neutrophils, as we saw before, they are filled with granules, so in the blood chapter. So the neutrophils are part of the blood, the leukocytes of the circulatory system, and they can wander in connective tissue, killing bacteria. Now, the neutrophils will exit the circulatory system when there is a chemical that attracts them to an area where there is injury. Otherwise, they don't have a reason to exit the circulatory system. And once they exit the circulatory system, they will go into the connective tissue, so outside of the blood vessels, and then they will try to find bacteria, and once they find bacteria, it will kill the bacteria. And then they can kill the bacteria by phagocytosis and digestion. So you will have within these granules these enzymes and bactericidal chemicals that will destroy these bacteria. Now, these specific granules are called the lysosomes, and within these lysosomes, they can have chemicals that are formed a response that is called the respiratory burst, and this zone around the neutrophil can destroy several bacteria at once. The eosinophils are found primarily within mucous membranes, like in the intestinal mucosa, and they guard against infections caused by parasites, but also they can be found in the respiratory mucosa. And if someone get in contact with allergens, many of you might have allergies, so these eosinophils will bind these allergens with an antibody is called that is called IgE. And when two IgEs that are found within the surface of the eosinophils are cross-bound by these allergens, these eosinophils will release several chemicals. Among some of them will be histamine, and then you produce this allergic response. So eosinophils will be important to kill parasites like tapeworms, roundworms, and they will use chemicals to kill these parasites like superoxide, molecules, hydrogen peroxide, and toxic proteins that will damage the nervous system of these tapeworms and roundworms. Similar will be for the basophils. Okay, so the eosinophils act similarly as the basophils, but the eosinophils can activate the basophils and the mast cells. And the eosinophils can phagocytose antigen antibody complexes. Basophils will encounter these antigens and then they will secrete chemicals that will help in attracting other leukocytes. The chemicals that they can release are leukotrienes, histamine, heparin. Leukotrienes will attract neutrophils and eosinophils, while histamine, no matter which cell it will be released from, histamine is a chemical that opens up the endothelial cells within the blood vessels. So this is a vasodilator, and this will increase capillary permeability. So you will have delivery of leukocytes into the area where they are needed, and also it will increase blood flow and plasma can exit the blood vessels because of this histamine. And heparin will inhibit the formation of blood clots that will impede the action of the leukocytes. Now the mast cells also secretes, besides the histamine, leukotrienes, and heparin, will secrete other substances that are important to activate the basophils. So the mast cells, it's a type of connective tissue cell that it is located in these connective cells but acts similar as to the basophils. Okay, let's talk about a specific action and functions of the lymphocytes. So the lymphocytes, as you saw in the intro of this chapter, were divided into T cells, T lymphocytes, B cells, and NK cells, or natural killer. Both the B and the T cells are only activated when you expose them to a specific antigen at a specific site area in the plasma membrane. So this is another key point for you guys to know, that both the T lymphocytes and the B lymphocytes are only activated by the exposure to a specific antigen and it's at a specific site in a plasma membrane. And then these lymphocytes will be subdivided in percentages. So the most abundant lymphocyte that we have will be the T cells, 80%. 15% will be B cells, and 5% will be NK cells. And the NK cells are part of the innate immunity, but can also be part of the adaptive immunity. And helper T cells will be functioning as well in both of them. Sorry. NK cells is only part of the innate immunity, while T cells can be part of both innate and adaptive, and B lymphocytes and the other cells will be part of adaptive immunity. Okay, so about the monocytes. The monocytes are basically the mother cells for the macrophages. So the monocytes will be found in the blood vessels. And when they exit the blood vessels, they will become macrophages within tissues. And the macrophages will be large monocytes that are avid cells for eating up debris and cells or bacteria. And the macrophages will be wandering just as the neutrophils in search for pathogens. We have fixed macrophages that are located always in specific tissues, and they have different names. So if the macrophages are located within the CNS, they will be called microglia. If the macrophages will be located within the lungs, they will be called dust cells, just like dust. And if the macrophages will be located within the liver, they will be called the Kupffer cells. Kupffer, Kupffer cells. And this is how the activated macrophages look like. This is a three-dimensional view of them with a scanning electron microscope at a higher magnification. You already saw a macrophage before, but that macrophage was attacked by, it was attacking bacteria. But in here you can see these couple of macrophages extending pseudopods. Okay, so along with those cells, let's talk about the antimicrobial proteins. So these proteins are called antimicrobial because they inhibit the microbial reproduction. Many bacteria reproduce very rapidly by binary fission every 20 minutes. So they can double their number very fast. And within our body, we have as part of this innate immunity, we have the production of these antimicrobial proteins. And these antimicrobial proteins will provide a short-term immunity against bacteria and viruses. And we have two major families of antimicrobial proteins. We have interferons and we have the complement system. The complement system will be around 30 subcomplexes of proteins that ultimately will assemble, and they form something that we call the MAC, or membrane attack complex, and they will follow in a specific way as they assemble. This complement system proteins are made in the liver. Now, interferons are produced by the cells that are virally infected only. They are not produced in cells that are not infected with viruses. So the interferons, they don't have any benefit to the cells that secrete them, because once the viruses infect the cells, they just replicate within the cells. But these cells, the ones that are infected with interferons, will secrete these interferons so to alert the neighboring cells so that they shut down the receptors on the plasma membrane so that they cannot become infected with viruses. I compare these virally infected cells and interferons as a good neighbor. Like, let's say that you get a call from your neighbor, and this neighbor tells you, oh, you know what, my house was broken in this morning or today, and I wanted to warn you that there is this burglar around the block, or of this event that happened to me, so that you can lock your doors or just check on your house better so you don't get broken in by this burglar. So this neighbor can be the virally infected cell, and then that message through the phone that is given to you is the interferon. And then you taking care of your locks in your doors and your windows, and then looking through the window, etc. That will be your response to prevent infection by the viruses, to prevent the burglary. Okay? So once interferons are received then by these neighboring cells, it will shut down more than likely these receptors, but also it will prevent, it will secrete proteins that prevents the infection by the viruses. So it will break down viral genes and prevents the replication of the viral genes. So you won't produce more viruses. And also this interferon is going to activate NK cells and macrophages. So in this case, this neighbor will call the police, the NK cells, and the macrophages and tell them, you know what, we had this burglary or one or two in the neighbor in the block, so take care of us. And then the NK cells will become activated and it will destroy these virally infected cells or these malignant cells. So the NK cells, the police cells, or the police will put this burglar into the jail. Now, for the complement system, as I told you, is made out of a group of 30 or more subcomplexes of proteins that will make a powerful contribution to both the innate immunity and the adaptive immunity. As I told you before, they are made in the liver, and then they are circulating in inactive form. So you don't always use the complement system unless you have a specific injury. So this complement system is activated only when you have a pathogen present in your body. Otherwise, there is no reason for activating the complement. Now, once you activate the complement system, you produce inflammation, you produce immune clearance, you activate your macrophages and your microphages, so you produce phagocytosis, and then you destroy cells, cytolysis or cytolysis. Cyto means cells, lysis means destruction. And then in order to activate the complement system, you use three pathways: the classical, alternative, and lectin pathways. These are complicated pathways that all of them, the classic, the alternative, and the lectin, they all merge into this dissociation of the C3 complex into C3A and C3B. You don't need to know so much about the cascades or the pathways, but DC3A and DC3B, they lead to different things. So the C3A leads to inflammation, while the C3B leads to immune clearance, phagocytosis, and cytolysis. And ultimately, no matter which the pathway it is, if you're going to have cytolysis, you will form a complex with C5b, C6, 7, 8, and 9, and these will form pores within the plasma membrane, which is this. This is a pore, and this will be the plasma membrane of a bacteria. So this will be MAC, membrane attack complex. So when you put a pore within the bacteria will leak its cytoplasm, and this is incompatible with the life of the bacteria. So the bacteria will become cytolyzed or dead. Now, the NK cells, the patrolling cells of our body, are always looking for anything wrong in our body, like in the search of pathogens or cancer cells, virally infected cells, transplanted cells as well. So once the NK cells find something wrong, they will bind into the abnormal cell, and then they will release these proteins called perforins. And these perforins, they will create a hole in the plasma membrane, and then this cell will die. Another thing that it can do is activate the cell, to have apoptosis or programmed cell death. So this is how NK cells work like. So this is an enemy cell. The NK cell will physically bind to the cell, will secrete these proteins that are called perforins. These perforins will form a hole in the cell, and then it will release these granzymes. Granzymes are enzymes that will induce cell death in this cell. So this is apoptosis or programmed cell death. And then once this cell die, the macrophage will come, the janitor cell will come, and then it will eat up this enemy cell. Okay, now let's talk about the other part of the innate immunity, the inflammation. So inflammation, it's a process that it is said when we have tissue injury or an infection. And this usually happens at a local level where we have that specific tissue injury. When we have inflammation, we are trying to prevent the spreading of microorganisms, spreading of anything that can further damage our body. And also, the inflammation helps to remove debris from the damaged tissue and also to start repairing the damaged tissue. An inflammatory response is characterized by 4 cardinal signs, which are redness, swelling, heat, and pain. Many processes in our body that are produced by inflammation will end in the suffix itis. So we can have arthritis, which is inflammation of our articulation, pancreatitis, inflammation of our pancreas, dermatitis, inflammation of our dermis. And many of these inflammatory processes happens because the cells that are injured releases cytokines. So cytokines are chemical messengers. And the cytokines are mainly secreted by the leukocytes, and they will alter the physiology of the cells that receives these cytokines. So you have this cytokine acting on neighboring cells, and this will be a paracrine signal. Or you can have cytokines activating their own cell, and this is called autocrine signal. Now, some of the cytokines will be like interferons, which will activate this neighboring cell. That will be a paracrine signal. We have interleukins, we have TNF or tumor necrosis factors, chemotactic factors, and others. There's a bunch of cytokines with different names, so we don't need to know them all. And once you have these cytokines released, you will mobilize the system of defense, and then you will start containing these pathogens from migrating to other areas of the body, and you start destroying these pathogens, and then you start cleaning up the tissue and repairing the damaged tissue. Okay, so one of the first cells that it is attracted by the cytokines into an area of injury are the neutrophils. So the neutrophils are found within the blood vessels, and once they sense these cytokines in the vicinity of these blood vessels, they will slow down their flow rate within the blood vessel, and then they will move into the side of the blood vessel to reach the endothelium. This is known as margination. And then within the walls of the blood vessel, we will secrete, or we will actually produce these proteins that are called selectins that will stop the leukocytes, so that the leukocytes will adhere to the endothelium, and then the endothelial cells will open up, and then the leukocyte will squeeze in between the endothelial cells in a process that we call diapedesis or emigration. So here is this neutrophil behavior in inflammation. If this was the skin of your finger, of your index finger, and then you got a splinter into your index finger, this splinter will create tissue damage in your epidermis and the dermis, which is this. So this is the epidermis, this is the dermis, and let's say this splinter has bacteria. Now, the tissue damage itself and the bacterial cells will help in the release of the cytokines. These cytokines will send a signal to these neutrophils that are located within this capillary, within the dermis, to slow down its rate of flow, and then to start marginating into the edge or into the wall of this capillary. This is the process of margination. And then your endothelial cells will open up, they will produce these gaps, and then you have migration or emigration or diapedesis. And then these cytokines will attract specifically these neutrophils into this area. So they will go into here and not away, because this is where you have the problem. And then your mast cells that are here also will migrate attracted by these chemicals. This process of attraction of cells by chemicals is known as chemotaxis. And then once you have these neutrophils coming into this area, they will secrete chemicals, enzymes that will destroy the bacteria, and then they will start eating the bacteria, and then they will secrete more chemicals to attract macrophages and set up an immune response that is more stronger than the neutrophils itself. And then afterwards, once you clear up this, you can start repairing the tissue. So what is the priority of inflammation? To prevent the spreading of pathogens. And then fibrinogen, which is a protein that is found in the blood vessels, it will form a blood clot that forms a sticky mesh that will wall off the microbes. And heparin will prevent the clotting at the site of injury, so that the pathogens are in a fluid pocket that it is surrounded by the clot, and then you can have attack of these pathogens by antibodies, phagocyte cells, and also other system or cells of defense. So how long it takes for the neutrophils to accumulate within the area of injury? Around an hour. And then they will set this response, immunological response, and then you will have a better protection. So this is the ways that neutrophils respond and kill bacteria: through phagocytosis, respiratory bursts, secreting of cytokines that will attract macrophages, more neutrophils, and the macrophages and the T cells will secrete this CSF factor that will stimulate the formation of more leukocytes, and you will have an increase in white blood cell numbers. So you will have neutrophilia between 5,000 to 25,000 cells per microliter in a bacterial infection. So at some point, one of the numbers in your differential complete blood count will be increased number of neutrophils in bacterial infections. Now, if someone has an infection caused by parasites, the person will have increased number of eosinophils or eosinophilia, and also the person can have eosinophilia when the person has allergies. Now let's talk about the second type of immunity, the adaptive immunity. So in the adaptive immunity, we have these three characteristics that distinguishes from the innate immunity. So the adaptive immunity is not local, it's systemic. So it happens throughout the body. And the innate immunity is local. The innate immunity, it is non-specific, so it will attack any pathogen the same way over and over again. And in the case of adaptive immunity, we have a specificity. So the attack is going to be specific against only one type of pathogen or a particular pathogen. In the case of adaptive immunity, and in innate immunity you don't have memory, but in adaptive immunity you have memory. So when you are re-exposed to the same pathogen, your body knows what to do. Now, adaptive immunity can be subdivided into cellular and humoral. Cellular or cell-mediated immunity will be that one that attacks anything that is wrong with the cells. So if you have a pathogen growing within your cells or infecting your cells, this cellular immunity will destroy them. And at some point, think about that T cells are part of the cellular immunity, while B cells are part of the humoral immunity. So, humoral immunity is also known as antibody mediated because B cells, B lymphocytes, makes these antibodies. And Greek doctors back in the times of Hippocrates and all those philosophers, um, or sorry, doctors, Greek doctors, they used to call the body fluids humors, like our blood, our urine, and all that. They call it humor. So anything then, any pathogen that is found or antigen that is found within your fluids, the body fluids, which will be CSF, cerebrospinal fluid, your blood, your urine, etc., it will be attacked by the B lymphocytes. Now, adaptive immunity can be further subdivided according to how you develop it into active and passive, and also into natural or artificial. So, active immunity will be when you produce your own antibodies, or well, when the antibodies are produced actually. So natural active immunity will be when you are exposed to an antigen or a pathogen and you start producing your own antibodies by the B cells, or when you start developing the memory T cells. So this is the type of immunity that most of us develop, because as we start crawling up and exploring our environment, once we're born, we start getting exposed to many antigens in the environment, and then you start developing your natural active immunity. And sorry, in both of them, natural active and artificial active, you are producing your own antibodies. So that's why it's called active, because you produce your own antibodies. Now, the difference between natural and artificial is that although in artificial active immunity you produce your own antibodies, it is only when you get artificially induced or exposed to antigens when you start developing these antibodies. Example of artificial active immunity will be vaccines. So in vaccines you have either an antigen or a pathogen, or an antigen from a pathogen, or an antigen, sorry, or a pathogen that it is partially dead or weakened or dead. So an antigen from a pathogen will never cause you an infection, similarly as for a dead pathogen. You will never get the infection. In attenuated, you might have if you have a very weak immune system. And then in artificial active immunity, you have to have booster shots, because at some point having only one shot is not enough to stimulate your immune memory. Now, in the passive immunity, you never produce your own antibodies. The antibodies, you will get them from either a natural source, which will be your mother, or an artificial source, which will be antibodies developed in a lab. So, natural passive immunity. The fetus will acquire antibodies from the mother through the placenta, IgG. IgG is the only antibody that crosses the placenta. Or if you were breastfed by your mother, you had received a second set of antibodies after you're born. In that case, that will be IgA. So who produced these antibodies? Your mother, but it will transfer you through the placenta, IgG, or through the milk, the breast milk, IgA. Now, artificial passive immunity. The labs, they produce and purify these antibodies, and they can be injected to you if you were exposed to rabies virus, if you were exposed to tetanus, if you uh, uh, if you were exposed to botulism, this bacteria that causes tetanus or botulism, or if you were exposed to a snake bite or a spider bite. Okay. Oops. So what are antigens? So antigens are any molecule, can be organic or inorganic, that triggers or sets an immunological response. Uh, we can have different molecular weights in the antigens. Some of these antigens can be small, some of them they can be large, but at some point the larger molecular weight of an antigen can be over 10,000 AMUs. And these antigens, independently of their size, can set an immunological response when your body does not recognize it as its own. It can be a large molecule, but it might not be as complex. It might be a simple molecule and your body recognizes as its own. It can be a small molecule, but if that small molecule has an odd shape or an odd structure, your body will not recognize it as its own. So, what can be kind of organic sources of antigens? Can be proteins, polysaccharides, glycoproteins, glycolipids, but at some point, uh, you can have inorganic antigens like dust, dander. Okay, and we have antigens that can be recognized as our own. These are self molecules. And we can have antigens that cannot be recognized as our own, which will be foreign antigens. And within antigens, we have specific regions that are the parts that sets this immunological response, which are called epitopes or antigenic determinants. It's like when the epitopes can be compared as when you don't like how someone tells you specific things. Like you can like a lot of things from the person, but in certain situations, the person might not tell you the things the way you liked it, and that can be the epitope that it can trigger irritability in you, if that makes sense. Okay, so haptens are small molecules that are very small that do not set an immune response. But when this happens, combines with a larger molecule, they can set an immunological response. So haptens itself then cannot set this immunological response, but when in combination with other things, they can set up. Example of haptens can be cosmetic detergents, industrial chemicals, poison ivy, and animal dander. Penicillin can also be a hapten. Penicillin itself will not produce an immunological response, but only when it binds to the host proteins is when it produces this allergic response to penicillin. So now let's talk about antibodies. So antibodies are proteins that are found in our plasma, and these proteins, these immunoglobulins, or these antibodies, are produced by the activated B lymphocytes or the plasma cells. Another name for these immunoglobulins are known as gamma globulins. And they are not only found in the blood plasma, but they also can be found in the tissue fluids after you secrete them from the circulatory system into the tissue fluids. Also can be found in body secretions like in the breast milk. In our tears we have IgA. In breast milk we also have IgA. We have IgA in the secretions of the intestines. In males, IgA can be found in semen. And also certain immunoglobulins like IgE can be found in leukocytes, like in the eosinophils. You can have IgE, or in the mast cells. Some immunoglobulins are found in monomers or only in one unit, but some of them they can be found in groups of 2 or 5. And the antibodies, they have usually 4 polypeptide chains that are bound by disulfide bonds. And you have 2 heavy chains and 2 light chains. The 2 heavy chains, they have 400 amino acids, and they have a hinge region where the antibody can bend and it can be flexible at some point to trap or to find antigens. The light chains are about half as long as the heavy chains, and within the tip of the light chains you have a region that is called the variable region that helps to be specific for finding specific antigens. And within this variable region we have the antigen binding site that attaches to the epitope. So this is so specific, this antigen binding site, that binds to a specific region of the epitope. And then you have also a constant region that is always the same in the part of the antibody that determines the mechanism of action. So this is the structure of the antibody. Looks like a letter Y. So this is the heavy chain here in green. Here we have the light chains. Here, these two, with these variable regions and this antigen binding site. So this is the place where the epitopes of the antigens bind the specific region. And then we have this hinge region which allows the rotation of these arms. So you can have flexibility to bind the antigens. And then here we have the constant regions that are always the same in the type of antibody, and these are the disulfide bonds that hold these amino acid chains. So the classes of antibodies that we have are 5, and they are named because they have a similar structure within the C region, the constant region. So we have the IgA, which is a monomer, so it's only found in plasma as a monomer, but you can find it as a dimer in the mucous membranes, saliva, tears, milk, intestinal secretions. This IgA will prevent the contact of pathogens in the mucosas and prevents these pathogens from penetrating these mucosas. They help to provide passive immunity to newborns if the baby was breastfed. IgD, it's a monomer, and it is found in the B cells. It seems to work as an activating factor for B cells once the the antigens bind to the B cells, so the B cells can become plasma cells and they can secrete other types of antibodies. IgE is a monomer that it is found in the surface of basophils and mast cells, and when you cross bridge two neighboring IgEs with an antigen, you will have a massive release of histamine and other chemical mediators of inflammation, and then you have allergy. And this IgE will help to attract eosinophils to fight against parasitic infections, and also will produce uh, the classical immediate hypersensitivity reactions or allergies. IgG, it's also a monomer, so you have only one unit, and it makes 80% of our circulating antibodies in the adult, and it will cross the placenta in the fetus, will be secreted as a memory type of antibody, and also it will help in the fixation of the complement system. IgM forms groups of 5, so it's a pentamer in the plasma and the lymph, and it is secreted when you have an immunological response, and it will create clumps of cells, and it will also help in the complement fixation, so basically in the attack with the complement system. So this is the pentamers of the IgM, and they can bind several antigens in different cells at once. In this case, the cells will be the red blood cells. So these antibodies, when they bind to the antigens, they will form an antigen antibody complex, as you can see here. So the ones in purple will be the antigens, and the ones in green will be the antibodies. So when you have these complexes will be heavy, and by gravity they will precipitate. So you have agglutination or clumping, and then you form a heavy weight that will drop or sink into the bottom of wherever they are located, and this will be called precipitation. Okay, so more details about the lymphocytes. So they are the major cells of the immune system, together with the macrophages and the dendritic cells, and they are located in specific places, strategic places that help us to prevent infections, like in the lymphatic organs that we talked about, the skin, and the mucous membranes. And again, the 3 types of lymphocytes will be the NK cells, the T cells, and the B cells. The lymphocytes, macrophages, and dendritic cells, they all help in concert to set this immunological response. So there are 3 stages in the life of a T cell. They will form in the bone marrow, they will train in the thymus, and then they will go into the circulatory system to carry out their immunological function. And then the training within the T cells in the thymus will be characterized by the release of chemicals that stimulates these T cells to mature when they are exposed to an antigen. And depending on the type of antigen that they get exposed to is whether they will be positively selected or negatively selected. So with the receptors that they have in the surface, the T cells will be capable of recognizing antigens presented to them, and then they will be positively selected. Now, when the T cells get presented with the antigens that you have in your own body, the self-antigens, but the T cells fail to react to the epithelial cell, and they react with the self-antigen, they will be negatively selected. So the T cells, they're supposed to recognize your own antigens and do not attack them. So if a T cell is presented with the self-antigen and they will attack them, it will be negatively selected. So you have to destroy them. So you will have a clonal deletion. It's like if you have a dog and you had it for several years and you work and you come at night, it is late and it's kind of dark. The dog, it bites you once after several years of having your dog, and you just talk to the dog and say, hey, what's happening to you? And then you just get into the house. But that happened again, and next time, and next time. Well, your dog, it is presented with the self-antigen, which is you, but it fails to recognize you. Will you keep your dog? More than likely not. So you will do negative selection. For as cruel as that sounds, but that's how our body works. And in some cases, you will have something that is called anergy, in which you have cell-reactive C cells that remain alive but unresponsive. So, negative selection will help you to leave the body in a state of self-tolerance. So you're supposed to recognize your own, but recognize what is not your own. And only 2% of the T cells will pass this test. So then within these T lymphocytes, once they are kind of trained, you will have a naive lymphocyte pool in which your T cells have not yet encountered foreign cells or foreign antigens. And when you have the deployment, your naive T cells will leave the thymus and will colonize the lymphatic tissues and organs everywhere in the body. And there, in those lymphatic organs and tissues, your naive cells will be responsible for finding foreign antigens. And if they find these foreign antigens, they will expand, and then they will form these clones that will attack these foreign antigens. Now for the functioning of your B lymphocytes. So your B lymphocytes will develop in the bone, and then some fetal cells, stem cells, will remain in the bone marrow, and then they differentiate into the B cells, and your B cells will start training within the bone marrow. So the B cells that react to self-antigen can either undergo anergy, like get half alive but are unresponsive, or clonal deletion, similarly as what we explained with the T cells. And whenever you have the self-tolerant B cells, the ones that do not react against your own antigens, they will divide rapidly and they will produce these immunocompetent clones. So you will undergo mitosis. And they will leave the bone marrow once they're trained, and they will go and colonize the same lymphatic tissues as the T cells. So both T cells and B cells, once they're trained, they will be located in the same areas. Now here's another point for your understanding. So your T cells and your B cells, they have to have this exposure to these antigens. And these antigens will be presented to the T cells and the B cells by the antigen presenting cells, which will be the dendritic cells, the macrophages, reticular cells, and some of the B cells itself. And here's the important point. The functions or the roles of the antigen presenting cells, this is a key point, will be to bind the antigens to the glycoproteins that they have in their surface, process these antigens, display it as antigen fragments, and activate the T cells. So one way that I compare this is when someone is going to get engaged. Many times they give this engagement ring in a box that it is wrapped with a gift wrapping paper. So in order for you to wear this ring, this engagement ring, you have to unwrap the box, take the ring off from the box, and put it into your finger. This is presenting your antigen. The part or the fragment, the part that it is important for you to activate the attention of people is to put your ring in your finger, your engagement ring, and show it to your friends. You're not going to show the box or the gift wrap, and you're not going to wear the ring. So you have to display this antigen in your finger and activate the attention of people by showing it, displaying this antigen fragment. So how does antigen processing happens? Well, your antigen has to be engulfed and presented by a macrophage. Your T cell will recognize the antigen. This is another key point all this slide. You will undergo several rounds of mitosis, so your T cells become differentiated into different types like cytotoxic T cells and memory T cells, and then your cytotoxic T cells will migrate to the focus of infection, and cytotoxic T cells will release perforins and lymphotoxins to get rid of the pathogen. And this is the diagram of the antigen presenting cells, how they engulf these bacteria. This is a macrophage. And then you have within this macrophage enzymes within the lysosomes that will fuse with this bacteria that it was engulfed through endocytosis, so it was phagocytosed. So you will form what we call the phagosome, and then these enzymes will start destroying this bacteria into pieces, and part of these pieces will be presented in this MHC protein, major histocompatibility complex protein, and then your epitope, your important part of the bacteria, will be presented in here, and it will attract the immune system cells. And then whatever is not important, it will be degraded and then thrown away. I mean, you can still keep the box for the engagement ring and the wrapping paper, but eventually you will put it somewhere else and you will wear the ring, the epitope. And then this antigen presenting cell, as it's presenting this antigen in the MHC, will attract the attention of the helper T cell. In the case of the person who was engaged, it will be the mom. First you say your mom, oh, you know what, I got engaged. And then your mom will call through these interleukins or cytokines the friends and say, wow, my daughter got engaged. And then it will get the attention of a bunch of immune system cells or your friends, the neighbors, and then it will be the news for the month. And then through these interleukins, you can have non-specific defense, humoral immunity, or cellular immunity. So if you attract the T cells, will be cellular immunity. If you attract the B cells, will be producing antibodies, humoral immunity. And if you attract macrophages, leukocytes, you start producing inflammation. This will be innate immunity or non-specific. And then this is how the T cells get activated. So you have your antigen presenting cell. Most of the times it's a lymphocyte, but a macrophage, but it can be a B lymphocyte. So your T cell, your cytotoxic T cell, will bind this epitope, and then it will release these cytokines, and it will undergo clonal expansion. So undergo several rounds of mitosis, and it will create differentiation into T cells, cytotoxic T cells, helper T cells, and then regulatory and memory T cells. And then through these cytotoxic T cells, you will destroy the cells, the enemy cells, and through these helper cells, you will attract other cells of the immune system, like NK cells, B cells, or more cytotoxic T cells. So cellular immunity, again, it's important for fighting bacteria, that it is infecting your cells or fighting foreign cells. And the immune system will remember specifically which antigens have activated the immune system. And then you will produce through this cellular immunity all the 4 types of T cells: cytotoxic, helper, regulatory, and memory. Cytotoxic will be the specific cells. That's why it's called cytotoxic, because cyto means cell, toxic, of course, will be damaging. So toxic will kill the cells. So that's why it's called cytotoxic T cells. Cytotoxic T cells will carry out the attack on enemy cells. These cytotoxic T cells are also known as CD8, CD8 plus, or T8. Helper T cells will be TH helps. They are important cells because they can become infected with HIV, human immunodeficiency virus. And once you destroy these helper T cells, which will be the ones that helps to promote the formation of more T cells and B cells, once they become infected with the HIV and they get destroyed, they don't make more of these cells anymore. So the only ones that we're born with will be the ones that we keep for life. So if they get destroyed by HIV, you cannot set up a strong immune response. So that's why they end up having immunodeficiency. And they help then to promote the formation of T cells, B cells, and innate immunity. Now, once you have controlled the situation, the immunological response is strong enough to prevent the spreading of bacteria or pathogens, you will use your T regulatory cells or suppressor T cells to inhibit the multiplication of other T cells and the production of cytokines. So it's like the ones that say to you, you know what, calm down, the situation, it's over. It is the cells that are very important. And like T cells, the helper cells, the regulatory cells can also be called T4, CD4, and CD4 plus. Now, memory T cells, they are derived from cytotoxic T cells, and they are important for developing memory. So when you get exposed to the same antigen for the second time, you don't set this immunological response from the starting point. So usually your primary exposure is called primary exposure to an antigen, and you take between 7 to 14 days to set it up. And once you have these memory cells, it will take like 3 days or so for setting it up again. So memory T cells are very important. And the stages of cellular immunity will be to recognize the antigen, attack the antigen, and then have a production of memory. And you can memorize this as the 3 R's of immunity: recognize, react, and remember. This is small T cells attacking a cancer cell. Now, the immune system through the humoral immunity also can help to attack antigens. Antigens can be neutralized by the antibodies, and it can tag them for destruction. When you neutralize these antigens with the antibodies, you prevent them from binding into cells. But also, these antibodies, they have in the heavy chains some sequences of amino acids that can be easily recognized by the complement system and by other cells of the immune system, so you can have a strong response. So memory and humoral immunity is because you will have differentiation of the B cells into memory cells, similar as for the T cells. So this is what happens then when you get exposed to antigens with the humoral immunity. So you have a bunch of antigens any body fluid. So you have these cells coated, these lymphocytes coated with antibodies. These antibodies will bind these soluble antigens, and then they undergo several rounds of mitosis. And then you will start forming these plasma cells that are going to secrete more antibodies, so you can bind more antigens in here. And then at the same time, you create memory cells. So for whenever you get exposed to the same antigen, you have memory cells that recognize these antigens very rapidly, and then you create a lot of antibodies to bind these antigens to neutralize them. So the antibodies have four mechanisms to attack the antigens. You neutralize them. So basically you block these antigens from binding into the cells. And then you can help in what we call the complement fixation, in which IgM or IgG can bind to the antigen, change shape, and initiate the binding of the several proteins of the complement system, and this will activate inflammation, phagocytosis, immunological clearance, and then cytolysis. And complement fixation will be important for detecting foreign cells, bacteria, and mismatched red blood cells. As well, the antibodies, especially the IgM, can bind several antigens at once, and it can create agglutination. And this will prevent the mobilization of more antigens. Now, as well, it will, as I said before, precipitate these antigens together with the antibodies, allowing them to remove by immunological clearance these antigens by the eosinophils. So all of these will be precipitating, and then the eosinophils can get rid of them. Okay, so one of the important points as well for the humoral immunity is that the first time that you get exposed to these antigens, your primary immune response will be to produce IgM and IgG. But IgM titers or levels are going to be initially higher than IgG in your primary immune response. Why? Because IgM is the first antibody that will help to bind these antigens. And then the memory cells will produce the IgG, but it takes a while more to make the memory cells. So that's why IgM is higher in the primary response than IgG. And when you get exposed for the second time to the same antigen, your IgG levels are going to rise sharply than the IgM. You will still have IgM, but at a lower level than IgG. And of course, this is important to understand, because one way to know if someone has developed memory against certain pathogens when the person was having this vaccination is by measuring the titers, the levels of antibodies. Like, many of you are going to work in the healthcare field, and you have received, hopefully already, or you might receive in the future, your vaccination against hepatitis B. So in your first shot of hepatitis B, your primary exposure, you get high levels of IgM first, and then you have still some levels of IgG, but not as high. And then in your booster shots, you have greater levels of IgG, but lower levels of IgM. And after you have received all your boosters, one way to know if your vaccination schedule was effective is to measure your levels of IgG. If they find high levels of IgG against against hepatitis B, that means that the vaccination was effective. If you find only IgM against hepatitis B, that means that the vaccination was not effective for some reason and that you need to have again your shots being given. And this is a diagram to show you how these levels look like. In the primary response, you have a sharp initiation. In your titers, you measure them in here at day 10, okay, on your primary exposure. So, or day 7. So you have high levels of IgM first, and then you have low levels of IgG. And then in your secondary exposure, if you have memory, you're supposed to have IgG, high levels, and low levels of IgM. Some immune system disorders, just to finalize, is that you can have a strong immune response to vigorous. So you can have allergies, or you can have autoimmune disorders in which you don't recognize your own antigens and you start attacking your own body, or you can have a too weak immune response because you're born with an immunological deficiency or you acquire it. And you can misdirect your immune system attack against the wrong targets in the case of an autoimmune response. The hypersensitivity or a strong immune response. This is the other name for allergies. In this case, you have an excessive immune reaction against the antigens that most people can tolerate. And you can have allergies to transplanted organs coming from a different person. This is called alloimmunity. You can have allergy or a reaction against your own tissues. This is called autoimmunity. Or you can have allergies to different environmental antigens like dust, mold, pollen, etc., including foods, drugs. And hypersensitivity or allergies can be subdivided into different categories, and they are numbered with Roman numerals. So the first one, it will be type 1, which is called immediate or acute, in which you have a very rapid response. Now you can have local type of type 1 and you can have systemic. Local will be like asthma, hay fever. It will be very mild acute response in type 1. If you have systemic response, this will be anaphylactic response. So you will have severe allergy throughout the body, and then you can die from there, from systemic hypersensitivity, which is also known as anaphylactic shock. This will be the type of persons who carry their EpiPen or their shot of epinephrine. Anytime that you have an allergy, that means that you were exposed before to the antigen. And this will be a secondary exposure to the same antigen, and you develop signs and symptoms to these allergies. Now, type 2 and 3 hypersensitivity are also known as subacute. They have a slower onset, so it takes 1 to 3 hours after the exposure to develop signs and symptoms. Type 1, it takes between 10 to, well, sometimes between 1 to 10 minutes to develop, sometimes up to 30 minutes to develop. But type 2 and 3 takes between 1 to 3 hours to develop the signs and symptoms. Sometimes it can last longer, up to 15 hours. And type 1, 2, and 3 are quicker antibody-mediated responses. Now, type 4 is also known as delayed cell-mediated response because it takes up to 3 days to develop these reactions or this hypersensitivity. Well, this concludes our chapter of the lymphatic system. Hopefully you enjoyed it, and if you have any questions, email me. And for some of you, have a good day or have a good night. Bye bye.